So welcome everyone to the NPTEL course on food science and technology. So this is basically the week 4. This course is basically instructed by Professor H.N. Mishra, who is the professor in the Department of Agriculture and Food Engineering, IIT Kharagpur, and I am Shikha Sharma, a research scholar in the Department of Chemical Engineering, IIT Guwahati, and I am the teaching assistant for this course for January 2026 semester. And this is our fourth live interaction session, and it is regarding week 4, and the assignment deadline for week 4 is basically 18th. So please do submit on time. So the topics that we will be discussing in today's class is basically includes module 4, which includes sensory quality attributes of food. So it includes significance of sensory organs, anatomy and functions of taste and smell, then sensory evaluation, subjective and objective methods, and then psychophysics of sensory perceptions, novel technologies in sensory evaluation like e-tongue, e-nose, etc., consumer acceptability. So it includes basically 5 lecture videos from lecture 16 to 20. So I hope you have all gone through all these 5 lecture videos. So if you have any doubt basically regarding this lecture videos, so you can feel free to ask me now. Also, in this live interaction session, we'll be solving some of the basic theoretical questions. It will be kind of a revising from these 5 lecture videos, the topics we'll be taking, and uh, we'll be revising all these topics. And I'm recording this video, so we can get this video at the end of this recorded video at the end of this lecture. So let's begin with today's session. So the first question for today's session is, which of the following senses is considered the first filter for food acceptance, influencing initial impressions? First is smell, second is taste, then third is sight, and fourth is touch. So which is the first filter for food acceptance? Anyone who would like to answer this question? Anyone who would like to answer this question? What is the first filter for the food acceptance? C. Hosea is saying C, which is sight. Sight is the first impression according to her. Any other answers? Okay, let's see answer. So she's correct. Sight is the correct answer. So we have basically 5 senses. So one is sight, another is hearing, then touch, taste, and fifth is smell. So basically eye is the first filter which tells about the food quality or the how food looks like or what is the appearance of the food. So first impression will come from the sight. So this is human eye. So from our eye we can perceive basically, or we can see the food. So sight is basically considered as the first filter for food acceptance because it gives the visual cues like the color, about the shape, and about the texture. So these are the basically initial indicators that people use to judge the quality and appeal of the food product. Then humans often make basically subconscious decision about the desirability of the food based on the appearance before even tasting or smelling it. For instance, a brightly colored fruit can be perceived as fresh and appetizing, whereas discolored or bruised fruits may be deemed as unappealing, or people may consider that it is a spoiled food product. So I basically process around 256 unique characteristics, which contributes to around 80% of human learning. I is basically capable of capture around 50 images per milliseconds, and that is too much of the images it can capture. And it enables the rapid assessment of the food attributes. While the smell and the taste impact significantly overall sensory experience, but they came into play only after the visual assessment. So they are the secondary or the tertiary uh, filters, but first filter is the visual assessment. Then touch is the secondary filter and contributes to the perception of the texture. What is the texture of the food product? So the touch, the smell, or the taste, they all will come into play later on. First will be the sight, how the food is looking is the first one. So sight is the first filter which helps to determine the quality or the if the food is safe. So it is the okay. Let's see second question. Which sensory organ is crucial for detecting food texture and mouthfeel? First is skin, second is ear and eyes, third is eyes and mouth, fourth is skin and mouth. So these are the different sensory organs which are crucial for detecting the food texture and mouthfeel. Anyone who would like to answer this question? Okay, D, I am seeing most of us are saying D. Okay, let's see. Skin and mouth. So you guys are correct. So basically, we have the five senses as we discussed already, that we have the vision, we have touch. So vision is basically for processing the what our eyes is seeing, okay, what our eyes are observing. So it is the vision is a process of perceiving the objects through eyes. Then second is the touch. In touch, basically we can touch any food product and can understand the texture of the food product. Then third is the smell. Smell is from nose. So our nose containing the nostrils which are used for breathing and smelling of the food product. Then fourth is the taste. Taste refers to the thought of the flavor which is felt in the mouth and the throat while we are having the food product. And fifth is the hearing. So based on the sound of like the food, like the chips are crunchy, so they produce some kind of sound. So based on hearing also we can sense the food product. So basically we have the different sensory modalities like the taste, which is related to gustation, and the basically taste is perceived by the tongue and the taste buds. Smell is basically we consider it into olfaction, and it is sensed by nose and olfactory receptors. Third is the sight. Sight is for vision, what we are seeing. So it is perceived by our eyes and their role in the food appearance. And then touch is basically we consider it into tactile modality and sensory modality, and it is perceived by skin and mouth feel. Then next is hearing, which is about audition. It is perceived by ears, and the what is the food is sounding. So basically, for food texture and mouth feel, we have skin basically and mouth. So basically, this is the for understanding the texture. This is under the touch or tactile, so tactile modality, and basically it is perceived by the skin and mouth. So D is the correct answer here. So basically the skin and mouth are the crucial basically for detecting the food texture and the mouth feel, which provide the overall or the comprehensive sensory experience. The mouth basically perceives sensations such as the creaminess or the crunchiness and the dryness, while the skin detects external texture related properties like smoothness or the roughness when food is being handled. So the combination of these sensory inputs, they will contribute to tell us what is the overall eating experience or what is the overall texture of the food product. Then eyes and ears, they are basically important also for evaluating the sensory characteristics, but they do not perceive directly perceive the texture or the mouth feel. Then next is the eye. So eye is for the visual clues or visual cues about texture, but they do not detect the physical sensation experienced during the consumption. So rest are incorrect, and option D is correct here. Okay, let's see this question. The sensory modality primarily responsible for detecting sensations like hardness and tenderness in food is. So which sensory modality we discussed about? There are different sensory modalities, like in this slide we discussed about these are the sensory modalities. So which one is related to hardness and texture of food? Gustatory, olfactory, tactile, or auditory? Which one? Please let me know. So Fausia and Shivaswamy are saying C, tactile property. Okay, let's see. So they both are correct. Tactile is the correct answer. So we have discussed that we have five senses like sight, hearing, smell, touch, and taste. So related to that, we have five different modalities which are visual, auditory, tactile, olfactory, and gustatory. So each modality basically respond to specific stimuli generated by the food product and has distinct neural pathways by which neural pathway they will carry these signals to the nervous system. So these neural pathways that will going to carry these stimuli to the central nervous system, which will form a broader sensory input. So if something is being perceived by the eyes, so eye will generate some kind of the stimulus molecules, and that will travel from the nervous system or the from the neural system to the nervous central nervous system, and the brain will going to understand the pattern and try to compare with the previous patterns and try to perceive what exactly it is seeing or observing. So this sensory input is then integrated with other incoming data and stored memories to access the overall food quality. So the tactile sensory modality, it involves the sense of basically touch. So it is responsible for detecting the physical sensations such as warmth, coldness, hardness, tenderness, smoothness, and crunchiness in the food products. So these attributes basically are perceived through mechanoreceptors in the skin and the mouth, which are basically are responsible for understanding or noting down the pressure, texture, and the force which is required to bite or chew the food product. So in this, we have hardness and the tenderness, which is generating some kind when we are biting the food product, so we can understand how much force is required through these mechanoreceptors. Next is the gustatory, which is refers to the taste. So it detects basically the flavor like sweet or the salty. Olfactory relates to the smell, which identifies basically aromas and the odors. And auditory perception, it involves hearing. So it is used to detect sound and like crunchy sound or the crisp sound, and but not the what is the physical texture or the firmness. So there are different type of tactile modalities which are there. So one is a pressure perception, another is a vibration perception, and third is the texture perception. So pressure, it basically involves sensing whether the food product is hot or cold when touched. Vibration perception is it detects oscillations generated during eating or drinking through sensors under the skin. Third is texture, which identify objects with varying levels of smoothness, roughness, or hardness using sensors in the fingertips. So here in this case, since we are dealing with the texture or the touch, it is tactile sensory modality. Next is the we have in column first different taste sensations like sourness, bitterness, salty, and umami taste. And what are the different in column two? We have the different corresponding compounds which are responsible for it, like MSG, monosodium glutamate, sodium chloride, caffeine, and citric acid. So we need to match these taste sensations with their corresponding compounds. So as we know in the mouth or in the tongue, basically the salt and the sweet taste is at the initially we perceive those taste. Then on the sides we have taste buds for sour taste. In middle we have for umami taste, and at the end we can understand the bitter taste. So we have the taste bud for bitterness at the end. So we when we eat something we'll feel bitter at when we are swallowing it at the end. So anyone who would like to comment on this question? So Shivaswamy and Forcia, they both are saying A. Let's see. So they both are correct. So basically tongue is the third critical filter in the food acceptance. So it allows basically to perceive a wide range of flavors. So it basically perceives human tongue basically detect five primary tastes which are sweet, salty, sour, bitter, and umami. So food texture, which is perceived through both touch and sight, significantly contribute to the overall taste experience. So all these properties like the basically touch or sight or other things, they taste, they all will contribute and will overall we can then tell what is the taste or what is the overall sensory perception of that food product. So taste and smell, they work very closely and making it difficult to fully perceive flavors when the sense of smell is impaired, such as when the snuffy nose is there. So basically taste and work together, they work. And if we, when we are having cold, we cannot smell the food product. So basically we cannot get the overall understanding of the basically the sensory attributes of the food product. So soreness basically is associated with the acidic compounds like citric acid. So it is basically on the sides of the tongue. Citric acid or the acetic acid or the lactic acid. So they can mainly found in the citric acid in the citrus fruits, which is responsible for the sour taste. And bitterness is basically linked to the caffeine, so which is a very strong bitter flavor, and it is present in the coffee and certain plant alkaloids. So it is bitter is the at the end of the tongue. So third is the saltiness. So saltiness is due to the presence of sodium chloride, which is the main source of the salty taste in the like common salt. From common salt we get NaCl, which is sodium chloride for salty taste. And salty is at the salt and the sweet is mostly at the like the initially we taste it. At the tip of the tongue, basically, we have the salty and sweet taste. And sweet taste is from the different sugars like glucose, fructose, or the lactose. Umami taste is the savory taste sensation, and it comes from the MSG molecule, which enhance the flavor of many foods by activating specific umami receptors on the tongue. So this umami flavor is basically found in the like meat, so it is meaty flavor we can say, and this is basically in the middle we of the tongue we perceive this flavor. So for soreness, citric acid; bitterness, caffeine; and for salt, sodium chloride; and umami, MSG. So A answer is correct here. So there are different chemical compounds which are involved in taste receptions like sucrose, which is for the sweet, umami for like glutamate. Bitterness के लिए molecule is basically chemical compound or the chemical structure is quinine. For salty NaCl and for sour citric acid. Okay, let's move to this question. Which of the following statement is true about the senses of taste? It is least important in the food perception. The tongue can detect only 4 primary tastes. Spicy sensation are felt as pain rather than the taste. All taste buds are evenly distributed on the tongue. So we need to tell which statement here is true. Anyone who would like to answer this question? D or C? We have two answers. Pradeep is saying C, Fozia is saying D, Shivaswamy is saying also C. Okay, let's see. So basically C is the correct answer here. So basically this senses of this spiciness is perceived as a pain, but it is not a taste because it detects by pain receptors like the nociceptors. So it basically specifically triggered by the compounds like capsaicin. So basically this uh, compound, it will basically trigger the pain receptors. It will not trigger the taste receptors. And this taste is basically felt as a pain. That's why. So this response activates heat and pain receptors in the mouth, creating a burning sensation in the mouth. So the sense of taste is basically crucial for food perception, impacting the flavor and the food preferences. And this statement is true. And since we have the five primary senses, we already saw that there are sweet, salty, sour, bitter, and umami taste. Uh, basically, primary tastes are there. So this statement is wrong. Then taste buds basically are not evenly distributed. They are having different concentrations in different areas, like this at the side and at the tip and the back of the tongue. They are different distribution. So it is also not correct. And first is saying it is the least important in food perception, which is wrong, because taste is very important for the food perception. So it is also wrong. So answer C is correct here. So let's move to this next question. Which of the following correctly describes how adaptation affects the sensory perception? First is increase sensitivity to continuous stimuli. Second is decreasing the sensitivity to continuous stimuli. Then maintaining constant sensitivity regardless of stimulus. And fourth is enhancing the perception of taste over time. So which one is describing the adaptation process or adaptation effect? Sometimes our taste buds gets adapted, right? So what is that? What happens in that case? So we have two answers as B, Shivaswamy and John. Okay, and Fosia also. So let's see. So they are correct. Adaptation basically is a sensory perception, refers to the phenomena where the sensory receptors they become less responsive to a stimulus when it is continuously present. So this lead to a decreased perception of the stimulus over the time. For example, if a person enters a room with a strong odor, they may notice the smell initially, but after some time, the intensity of the odor seems to diminish as the olfactory receptors adapt. So, answer B is correct. Okay, let's move to this next question, which is regarding the taste buds. So, which of the following taste buds are responsible for detecting the sweet taste? First is the foliate papillae, second is the circumvallate, third is the fungiform, and fourth is the pyrenal. So, which taste buds are responsible for detecting the sweet taste? Fungiform papillae, Fausia and Pradeep. Okay, let's see. So they both are correct. So this is our uh, tongue in which there are different taste buds present at different locations. So fungiform are here, then folate after that, circumvallate are there. And in these papillae, we have the taste buds. So the taste buds basically are the peripheral chemoreceptors which are located on the dorsal surface of the tongue. So some are basically also found in the soft palate, pharynx, and these are the different parts, basically smaller parts of the in the tongue, larynx and upper esophagus. So these are the different parts. So taste buds are basically the chemoreceptors. They are basically the chemoreceptors which basically take the whatever is the taste is there. So based on that, the receptors will activate and send signal to the central nervous system. Each taste buds, they consist of around 30 to 50 modified epithelial cells clustered together in a layered ball-like structure. So these taste buds basically are housed in specialized structures which are called papillae, which forms the bumps and the grooves in the tongue. So we have in the tongues, our tongue is not very smooth; it's little rough. So that is because the taste buds basically uh, are present in the papillae. So because of that, our tongue is having some bumps and the grooves, and these are basically responsible for understanding the taste. So fungiform basically they are located primarily on the in the basically front part, anterior part of the tongue, and are responsible for detecting the sweet taste and the salty taste, also sometimes umami flavors. And they are the mushroom shaped structures that contain multiple taste buds, making them sensitive to a variety of taste stimulus. So another is the foliate. So the foliate is the sheet or the folded like structure, and they are located in the side of the tongue, which are responsible for the detecting of the sour and the salty tastes. The third is the circumvallate, which are like the tower-like structure, large size papillae at the back of the tongue, and they detect basically the taste. And last is this is the taste bud, pharyngeal. They are basically the taste buds which are located in the throat area, and they play a role in the evaluating the quality during ingestion. So all of these helps in understanding the um, food taste when we are swallowing. So basically, for sweet taste, the first is fungiform, basically important. So answer C is correct here. Fourth is we have in column 1 different taste disorders, like we have 4 taste disorders, and in column 2 we have their descriptions. So we need to match here these. Anyone who would like to answer this question? I hope so far is clear to everyone. Whatever we are discussing is clear. B or D. We are confused between B and D, right? We have two answers, B and D. Okay, let's see. So answer D is correct here. Ageusia basically it is the taste disorder where there is a complete loss of the taste sensation is there. So there is no taste perceived at all. So This is related to this one, which is complete loss of the taste sensation. Then hypothesis is basically the condition where there is a reduced ability to perceive the taste, where the taste intensity basically is lesser perceived. So second one is related to C. Then third, which is dysesthesia, is basically is related to the distorted taste perception, where we can have some unpleasant or metallic or bitter flavors we can perceive. So it is related to B. Then fourth one, which is pantosia, is basically related to the perception of the taste without any stimulus. So we can perceive the taste. Individuals, they can perceive the taste even if they didn't have, or they don't have, they're not eating any food or any beverages. Still, they'll perceive some kind of the taste. So fourth one is related to D. So answer D is correct here. Also, there are two other problems or disorders related to taste, which are glostis, which is the inflammation of the taste buds, basically, or inflammation which can alter the taste. And another is zero, there is a dry mouth, and it is often caused by medication or medical condition. The sense of taste is also sometimes affected by the loss of the smell. So we can have taste buds and we can have smell. They both will act and give the perception of taste. So if that in the taste bud we have some kind of problems, so we cannot have the proper taste of the food product. We can feel the bitter taste, metallic taste, or we cannot even perceive the taste because of presence of all these disorders. So in this, answer is what? I hope this is clear. Let's move to the next question. So which of the following describes the role of basal cells in the olfactory epithelium? First is they bind odor molecules. Second is they secrete mucus to trap odorants. Third is they regenerate damaged olfactory receptor cells. And fourth is they transmit signals to the brain. Anyone who would like to answer this question? D and C. They secrete mucus to trap odorants. Basal cells, okay. C is they regenerate damaged olfactory receptor cells. Okay, so we have two answers here from Shivsami and Fozia. Anyone who would like to answer or tell which one is correct, D or C, or any other option, A or B? Okay, let's see the solution if no one is have the answer. Okay, so C is correct in this case. So this basal cells, they are basically used to regenerate the damaged olfactory receptor cells. So if we see our nose, basically, so it has nasal cavity here. This is the nasal cavity. So it is basically the large air-filled space located behind the nose, and it is basically divided in two sections by the nasal septum. So this is the just behind the nose. We have this large air-filled spaces. So these are known as nasal cavity. Then second is nasal passage. So that is basically the superior, middle and inferior nasal turbinates, which increases the surface area of the nasal cavity. This design of the nose or the nasal cavity, it enhances air filtration and improves the detection of the odor. Then next is the olfactory region. So in that it is basically situated near the cribriform plate of the ethmoid bone in the upper nasal cavity. So it is somewhere in the upper nasal cavity here around. And this area basically contains the olfactory epithelium, which is responsible for detecting and initially processing odorant materials essential for the sense of smell. So it is this olfactory region is this, the nasal cavity is this, and in between is a nasal passage. So this olfactory epithelium, they have basically three type of the cells: olfactory receptor cells, supporting cells, and the basal cells. So the olfactory receptor cells, they basically have the specialized sensory neurons with cilia like the hair-like projections. So these are basically the receptor cells, olfactory receptor cells, this one. Okay. So this is basically having the cilia, which is hair-like projection. So they are extended in the mucus. So this is the hair-like structure in the mucus. So when odor molecules will come and bind to this receptors on these cilia, they generate some kind of electrical signals which are transmitted to the brain via the olfactory bulb. So from this epithelium, these cells, basically the electrical signals will generate, and from these optical nerves they will basically go to the brain. Then supporting cells are basically to provide the structural and metabolic support to the olfactory receptor neurons. So these are basically, this round one is the supporting cells, so to provide the structure to this olfactory receptor neurons. Then third one is the basal cells. So these are basically olfactory epithelium in the basal cells. They are basically in the olfactory epithelium, and it functions as a stem cell. So stem cells, what it do, like stem cell, it functions. So they basically regenerate and replace the olfactory receptor neurons, which are continually damaged or lost due to the exposure to the environmental factors like the pollution or coming of the pathogen. So they have the regenerative capacity, which is very important because olfactory receptor cells, they have the shorter lifespan and they need constant replenishment. So these olfactory receptor cells, they have the short lifespan. So basal cells will going to help in regenerate these cells. So this blue one, they are the basal cells. In this question, basal cells, they are related to regenerate the damaged olfactory receptor cells. Okay, so let's now the next is sensory evaluation test we'll discuss. So which sensory evaluation test is most suitable for routine quality control to ensure product consistency? Hedonic test, descriptive analysis, do your trio, or triangle test? So we have two type of the test techniques. One is the analytical or objective test, so which is related to the product oriented tests they are. So it is about the quality or the quantity of the product characteristics and the similarity and dissimilarity between the products, or the standardization, or the basically the trained persons or sensory evaluators, they will evaluate the food in this. So it is classified in discriminative or descriptive. So, and second is affective test or the subjective test, which are people oriented tests. So whether the people have the preference for that food product, or will they accept the food product, or what is the first impression from the people, or the what is the like after eating, what the consumers will going to say about the food product. It is all about that. So next is the large number of the panelists and representation of the people population. So it basically it is given to the different people, and so that we can try to understand how the food is being perceived by people. It is not about, and in analytical techniques we analyze the food product quality, and we understand from the perspective of the product. So affective tests, it includes hedonic, scale, acceptability, and preferences. So in this question, we are talking about which sensory evaluation tests, which includes both the affective here and the analytical. So which is most suitable for routine quality control analysis to get the product consistent. Anyone who would like to answer this question? A, D. Okay, D is the answer by Shivasamy and Fosia. Triangular test, they are saying. So they are correct. Triangular test is the most suitable for testing on the routine basis. The quality analysis is done, and the most suitable is triangular test. So after this, we have seen that there are two methods, like the discriminative and descriptive. And here is the hedonic, acceptability, and preference. So in the discriminative test, this triangular test comes. Also there are different tests which comes under this discriminative test, like the paired comparison test, triangular test, then duo trio test, and alternative forced choice methods, and A not A test, sorting methods, ABX discriminative tasks, and dual standard tests. So there are different tests which comes under the analytical test, under especially the discriminative tests. So this analytical or objective tests, they are also known as discriminative tests, and they are used to determine whether a perceptible difference exists between the samples or not. So product developers, they often use this method when they are reformulating the products with different ingredients, aiming to ensure that the consumers do not notice any change between the two products. So suppose one product is uh, anything like the fried snacks, maybe we can say samosa. So there is a fried snack, and if we are trying to make the samosa again, but with the healthier ingredients, maybe with the help of millets, or maybe with the help of other things, so uh, s- we want that product should be equal to the like the taste will should be same in the healthier version as compared to the existing one. So there we can use this test. So panelist personal preference, like or dislike, do not affect basically the outcome, as the test focuses purely on detecting the differences of the product. Typically, trained personalists basically are used here in this test, while the test can identify if a different exists. If uh, it doesn't quantify basically the degree or the nature of the difference. So the trained personalists are basically used here so that they can identify if some difference is there or not. So in the triangular test, basically, it is most suitable for the routine quality control because it is a quick and effective method for determining whether a perceptible difference exists between the two batches or not. So suppose there is this batch 1, 2, and 3. So we can understand by the trained personalist, what we can do. We have these three samples within which two are identical and one is different. So the personalist can identify the odd sample, which one is the odd sample, and which two are similar or identical products. So this method basically helps to detect even subtle differences which are arise from the variation in the ingredients, processing, or storage, ensuring that the product consistency should be there over time. So the basically the results from this are analyzed based on the probability that if there is no difference exist, the odd sample will be chosen by chance of 1 by third. So as per the probability equals to 1 by 3 for each, right? So here we can understand from the trained personnel will come and try to understand which are the similar products, identical products, and which one are the odd one product in this triangular test. Next is the hedonic test. In that there is a 9-point hedonic scale is there. So that measures basically the consumer preferences and likings, which is basically not possible by the routine quality test. So it's not possible to always take the, like always measure through the hedonic test. So the simpler and the more practical for taking the routine test is triangular test. Then next is duo trio test, which is again comes under discriminative test. Hedonic basically comes under this affective or subjective test. So duo trio test, they also determine the differences, but it is less efficient than the triangular test for quality control purposes, because here what happens, we will have one reference given to the panelist and two coded samples we will give A or B, and the panelist will going to identify which code of the sample will going to match with the reference sample. So here, so the null hypothesis states a 50% chance of selecting the correct sample by chance. So probability of selecting the correct from these two set of samples will be half. while the alternative suggests a higher probability if a difference exists. So if there is some difference will be there, so there is a chance that the most close sample will be chosen in this case. So it is preferred for strong flavored samples and detect the difference without specifying their nature like salt, sweet or sour. So if there is a strong flavors are there, so do your trio test can be done. So if we see the triangular test, in the triangular test, we need to taste each sample and identify which one is different. So we have three samples, suppose we'll have three samples and we'll say, the trained persons will going to taste these three samples and we're going to say which two are identical and one is different. In do trio we will, the trained personalist already will going to like know that there is this reference and which one is close to the reference, either A or B. And third is the ranking test, in which there is the like we'll have the panelist will have different set of the samples based on the intensity of the flavor, so that he'll going to arrange in the order from like less intensive flavor, then more, then more, and then more. So he'll going to rank the samples in the increasing order of the flavor intensity. That is the ranking test. So right now we were discussing about the discriminative test. Now, what is a descriptive test? So in descriptive test, basically, it provides basically detailed and quantitative information about specific sensory attributes. In discriminative test, we were just qualitatively we were telling that this is more close to this sample or more close to the reference. We were not quantitatively determining, but in descriptive analysis, we're going to have some number or some quantity basis. Will be gonna analyze this sensory attribute. But this method is time consuming and it requires highly trained person or panelist. So that's why it cannot be done on the routine basis. Only discriminative techniques we can do on the routine basis. So in descriptive tests, they are basically help us to understand how changes in formulation or the processing or the storage it affects the sensory profile of the product. It can be used for product development, quality control, benchmarking against competitors, etc. This analysis can be used in difference testing and in product development. Also, the intensity of each of the described attribute can be quantified by this method, and it provides a complete basically discrimination of the sample difference and guides the product developer in modifying the product characteristics to meet the consumer demand. So it involves basically trained personalist will be there 6 to 8 who will going to systematically assess the intensity and the quality of specific attributes in the food sample such as the flavor or the color, etc. So in this, we'll have highly trained persons who will going to quantitatively tell. They'll give some numbers or the ratings, which will be based on the quantity or the intensity of the color or the flavor. So basically, triangle test is the best choice for consistent and effective monitoring of the quality of the product. So in basically descriptive analysis, we can have some numbers and we can overall perceive the sensory perception, like we'll have some numbers and we can overall tell, okay, this is the final number. So we can compare between different products and can understand the intensity of that particular flavor or the color. Okay, let's move to this next question. Which of the following statement are correct with respect to the affective testing in sensory evaluation? So statement 1 is saying it is conducted with trained personnel to measure consumer preferences. Second is it is not provided it is not providing the quantitative data for statistical analysis. About the affective testing, this one, which statement is correct? Anyone who would like to answer this question? So how many people today joined? Okay, so answer Shivaswamy A, Fozia D, neither one or not two. Both statements not correct. Let's understand this question. So basically this affective testing basically is used to measure the consumer preference, but it is conducted with 50 to 100 untrained personnel such as regular consumers and not non-trained persons. So basically it is conducted with the help of untrained persons. So because we need to here, the purpose is to understand how people will like or dislike the product based on their personal preferences. So we will randomly can go to the consumers and try to understand whether the product they are liking or not. So it is not conducted with the trained personalist, which is statement 1 is wrong. And second is that affective testing does provide quantitative data. So they provide the quantitative data for statistical analysis. So which is often using like scales like hedonic scale, 9-point scale to gather measurable and analyzable response. So basically this statement is also wrong because it provides the quantitative data for statistical analysis. So both of that statement are incorrect. So neither one and nor two is the correct answer in here. Answer D is correct here. Nor this is because it provides basically the quantitative data for statistical analysis like 9-point hedonic scales are there. So we can measure and we can analyze the response based on the stats. So D is correct, neither 1 and 2. So basically this allows researchers basically to apply the statistical techniques to determine trends and the preferences among the consumers. So we can ask panelists the questions like which product do you prefer? Untrained panelists, we can ask questions such as these, like which product do you prefer, which product do you like, how will you do uh, if you like this product, and how often will you going to buy this product or use these products. So the consumers are basically the final judges here for purchasing and eating behavior. We can judge based on the consumer's perception and the location and the subjects, and we can have the different test designs and questionnaire formats. So these are the different parts we can use in this testing process. And three types it includes, like we discussed: preference test, acceptance test, and hedonic scale. So hedonic scale is a 9-point scale, and preference test, they allows consumers to express a choice between the sample where one is preferred over another or no preference is indicated. And in acceptance test, it is used to understand the degree of consumer acceptance for the actual use or purchasing or eating for the product. So this category test, pair comparison test, ranking test, they are basically used to assess the product acceptance. So in preference test, we'll have the competitor product and our product. So we'll see, ask the customer which will you going to prefer. And in acceptance test, if they are actually using the product, so how much or the degree of the acceptance by the consumers is being analyzed. So here answer is D, neither 1 and 2. Okay, let's move to this question. So column 1 is having different tests like triangular test, descriptive analysis, hedonic testing, and paired comparison. In column 2, we have different tests which have the corresponding description, like measuring consumers' liking or preferences, describing the sensory attributes in details, then identifying difference between the samples, and then comparing two samples to determine preferences. We need to match this. So Shivaswamy and Fozia, they both are saying B here in this case. Okay, so they both are correct. So basically descriptive analysis, they involves detailed characterization of the sensory attributes of the product, usually by trained panelists, and provide a comprehensive profile of the flavors, texture, and the aromas. And we already discussed about the descriptive tests. So this descriptive analysis is related to this. In detail, we are understanding, because in triangular test, basically it's a discriminative method which is used to identify the difference between the samples. So there two will be identical and one is different sample, and we can find odd one out. So this test is basically for assessing the subtle difference in the sensory attributes. So first is related to C, that we identify the differences between the samples. Then hedonic testing is basically measures the consumer liking and the preferences, typically using untrained consumers who rate their enjoyment on a product on a scale. So it is used for understanding the market acceptance. So hedonic test is related to the consumers or the untrained panelist liking. And paired comparison test basically is one kind of preference test, as we discussed here, that in preference test here we can have in preference test, which is basically comes under the affective test. The preference test which is come under the affective test, in this the paired comparison test comes. So paired comparison is a preference test where the panelists are presented with two coded samples. So panelist will have sample A, sample B, and we need to ask, or we need to indicate which one they prefer, the panelist which is going to prefer from one or two. So making it suitable for direct comparison between the two products. So suppose we have two samples A and B, okay, and it will be in identical containers, and they will be having some 3-digit random codes. So that there will be no biasness. Biasness can be avoided. Should not be there. So we will put in same container with random codes. So two possible orders in which we can give, either first we can give A, or second we can give B, or first we can give B and then second we can give A. So each order should be presented an equal number of times. So sample must be evaluated from left to right, and retesting of the sample is allowed. So we'll give this A and B two samples, the same samples again and again to the panelists in the same equal number of time we'll give. Like if I'm giving A in the first place and B in the second place, then I'll give B again 3 times in the first place and A in the second place for again 3 times. So there will be retesting allowed here, and we can give, there will be no biasness if we will going to do this. In this there are two different things, which is simple difference test and directional difference. So in the paired comparison only. So in simple difference test, panelists basically they are asked to test whether the sample in each pair are the same or different. And whereas in the directional difference, the panelists basically they asked to indicate which sample in the pair has greater or lesser degree of intensity of a particular sensory attribute. So the format for this evaluation form can be like this. For simple difference, that we will have two samples A and B. So they can tell which sample they have the different one, or which sample is the same, like this. Different or not different judgment is in this. Whereas in directional difference or the preference, so they'll be having, like, we'll have different intensities. We need, panelists should tell the intensities also, and the form is like this. And these type of the, when we get this from the form, we'll going to get the data. So the data will be analyzed using binomial or multinomial distribution probabilities, like this, that number of judges which are preferring the sample X divided by the total number of the samples, like that, in the based on the probabilities or the binomial and multinomial distribution. These will make the table and we're going to analyze. For panel selection, product differences or preferences, we're going to use this distribution. And when there is number of the observation which will exceeds the table value, chi-square test we can use t-square for percentage of product difference or preference. So basically later on these data can be analyzed with the help of the binomial and multinomial distributions and chi-square test or t-test. So this is the paired comparison test, which is comparing two samples to determine the preferences. So here answer D is correct in this case. So your answer B is correct in this case. 1 is related to C, second is B, 3A, 4 D. Okay, let's move to the next question. Which test is used to determine consumer acceptance of a product, asking panelists to rate the product on a 9-point scale? Triangular test, hedonic, discriminative, or descriptive? Anyone who would like to answer this question? Shivaswamy and Fozia B, hedonic scale. So you guys are correct. Hedonic scale basically is a sensory evaluation method which is used to determine the consumer acceptance and preference of a product. In this test, panelists will going to be asked to rate how much they are liking or disliking a product based on a 9-point scale. So it will range from like extreme liking to extremely disliking. So 9, if someone is rating 9, that means it can be extremely liking, like the product extremely, and 0 means not liking. So the hedonic test is basically designed to measure subjective preferences and the overall liking, making it an essential tool for the product development and consumer research. So it is this hedonic 9-point scale sensory evaluation methods is very important in the research area also when we are developing some food product, or in the industry also it is basically used. So when we are developing some food product, we go to the random or the untrained persons, and we will going to give them the product, they'll test, and they let us know if they like the product or not like. So in this basically what we do is description of the panelist task. So panelists basically are asked to evaluate the coded samples we'll give to the panelist, and they're going to uh, basically taste and evaluate and rate their liking or disliking based on a 9-point hedonic scale. And we're going to basically how we're going to present the sample is we'll give 3 or more samples, so that, and we'll give in the identical containers which will have some unique codes or random numbers. So the sample, they'll be presented to the consumers in the random order or the balanced order, basically in the same container, and with the codes we'll going to give for the evaluation of the food product, or the for the sensory evaluation of the food product. So this is the kind of form we can make where we can have the participant name and participant number, and this like this. There is a different samples, like 4 samples are there. We can give some code, and we can have different attributes like color, texture, aroma, taste, overall profile. Or we can add mouth feel also here. Mouth feel also sometimes. So on the consumer will going to like, if like, so they'll give 9, and if they don't like, they'll give maybe 0. Extremely. So here is the scale. So if they like extremely, they'll give 9, and if they dislike extremely, they're going to give 1 mark. So like this sensory evaluation based on hedonic scale proceeds. Next is the triangular test. So this is basically a discriminative method which is used to determine if the difference exists between the two products or not. So here there is no meaning of liking. And in discriminative test, it identifies whether there is a perception difference between the samples but does not access preference. And in descriptive test involves basically trained personalist providing the detailed description of the sensory attributes of the food products and does not measure the consumer acceptance. So here is a hedonic method which accepts a consumer acceptance and on the 9-point scale. Okay, let's see this question. Which of the following is essential for minimizing the biasness during the sensory evaluation? So first is using clear and descriptive sample labels, then conducting test in a communal environment. C is randomizing sample presentation order. Fourth is allowing panelists to discuss their preferences during the test. So which one is going to contribute to the minimum biasness when we are doing sensory evaluation? Shivaswamy C, randomizing the sample presentation order. Pradeep is D, allowing panelists to discuss their preferences during test. And Fozia is saying C, randomizing the order. Okay. So C is correct answer. That randomizing the order of the sample presentation, if we randomize the order of the sample presentation, we can reduce the biasness or we can minimize the biasness during the sample evaluation. So when the sample is presented in the random sequence, it prevents order effects. So if the sample is in random, so it's not like that like we are not putting on the based on the increasing intensity or decreasing intensity, so order effects can be prevented, such as a panelist developing a preference or aversion to the sample based on their position. So based on their position, sample panelist can have some mindset, but if we are giving randomly, so biasness can be reduced. For example, if highly flavorful sample always follow a milder one, the panelist might perceive that the second sample as more intense due to contrast. By randomizing the order, each sample is judged on its merits, ensuring a more objective evaluation. So using clear and descriptive labels could introduce biasness. So if we are using the clear or descriptive labels, we are telling the panelist that in this sample this product is there. If we are using the clear labels, not the coded one, so that can increase or that can contribute to the biasness. Also, if we have the communal environment, so it can lead to group influence or the biasness. If we are allowing the panelists to discuss their preferences, that can lead to the conformity, and peer influence also can be there. So that can skew the results, that can change the results. Biasness will be there. So C is correct, that we should randomize our sample order to get the minimum biasness, and we should use always the codes, and we should always do this sensory analysis on an individual basis, not the community basis, and we should not allow the panelist to talk about their preferences. So one person cannot influence, one person's mindset cannot influence the another person's preferences. Okay, so there are different sensory evaluation errors like halo effect, order effect, expectation errors, and leniency errors. So in column 2, they have the description given, like panelist expectations influence perceptions, previous sample affects the evaluation of the current samples, rating one attribute influences ratings of other, and panelists tend to give more favorable scores than warranted. Which one is anyone who would like to answer this question? Okay. So you both are correct. C is the correct answer. So the halo effect is basically occurs when the panelist rating of one attribute influences their perception of other unrelated attributes, such as appealing aroma, leading to higher ratings for taste and texture. Order effect is describes when there is a how the evaluation of the sample is influenced by the one tested immediately before it. That is the order effect, making subsequent samples seems stronger or weaker in comparison. Then expectation error arises when a panelist have some preconceived notions such as the product labeling or prior experience. So it can affect their perception, leading to the biasness in the ratings. And leniency error means that there is a tendency of the panelist to give more favorable scores than justified, often to avoid being overly critical or to maintain a positive evaluation atmosphere. These errors emphasize the importance of controlling the biasness while doing the sensory evaluation to ensure accurate and reliable assessments. So we have different psychological factors which affect and produce the error, which is expectation error, suggestion, then stimulus and logical error, distractions, order effect, attribute dumping, and halo effect and proximity error, central tendency error, contrast and convergence error, motivation error, and acclimation error. So these expectation error, they basically information about the sample or objectives can influence the panelist to find what is expected. Suggestion error means when conduct evaluation in a controlled environment. So to avoid if the panelist can hear some comments or sound or suggestion from another, they can have this suggestion error. And stimulus and logical error occurs when the evaluators associate the stimulus with the unrelated attributes, leading to the biased decisions. Distractions can be there with the radios or the chattings, which can lose the panelist focus. And order effect means when the response to one sample may influence the perception to the next one based on the positioning. They can have some biasness. Attribute dumping means ensures the sensory evaluation from covers all attributes to avoid incomplete assessment. So halo effect means and untrained basically persons, they judge the multiple traits at once. So they can be different, like one attribute can influence the other attribute, like the color. If the color is bright, the untrained person can think that its taste will also be better. Then central tendency error, untrained evaluators, they lead to give scores in the middle, basically like they'll give average score like 4 or 5 to all the samples in the middle of the scale when using rating system. Contrast and convergence effect, it reduces these effect by randomizing or balancing sample presentation and eliminating outlier from the sample set. So this can be reduced by randomization. Then motivation error, panelist may rate sample based on their feelings or their emotions towards the manufacturer or the company employees. Then acclimation error occurs when the judge repeatedly evaluate the similar items. So minimizing this by introducing spiked sample or varying the product. So if the sample is already in the past is judged by the panelists, so they know already that type of sample. So because of that, some error or biasness can be there. So in this question, halo effect is basically related to the rating influence of the one attribute over other. Second, order effect is because of the position, and expectation error is because of the panelist expectations, and leniency error is more giving more scores than needed. Next is what is the factor to for that we can minimize the central tendency error in sensory evaluation. Training personals to use the full range of the scale and conducting evaluation in a noisy environment using only untrained persons and presenting all samples in a fixed order. So which one is related to minimizing the central tendency error? Anyone who would like to answer this question? A from Shivaswamy, Fozia and Pradeep. All three are saying A. Okay, so you guys are correct. So central tendency error, it occurs when the panelists basically avoid giving the extreme scores and they try to give the score which is in the like and tend to cluster their ratings around the middle of the scale. If they are giving like they are not giving 1 or they are not giving 9, mostly they are giving 4 or 5 in the hedonic scale, suppose. So that is the central tendency error. So this error can distort the results and make it difficult to differentiate between the samples. To minimize this error, it is crucial to train panelists basically to use the full range of scale confidently and appropriately. So ensuring that they give higher or lower scale when the samples deserve that. So we need to teach them that if they are giving like 1 or 9, so they can give and uh, based on the performance of the sample, they can award the marks. Then conducting evaluation in a noisy environment, it can even introduce the distraction and negative impact, and the accuracy of the sensory can go down, and using the untrained persons only, it increases the likelihood of the central tendency error as they may unsure about the using the extreme rating. So we should use some trained personalist. Then presenting samples in a fixed order, it can introduce the order effect. So another source of the biasness will be there, but it does not directly address the central tendency error. So, so answer A is correct here, that we should train the personalists to use the full range of the scale. Next question is which of the given option is correct with respect to the effective practices for determining the recognition threshold in a sensory test using forced choice method, providing detailed background information about each sample, starting with concentration well above the expected threshold and avoiding distractions in the sensory environment. Anyone who would like to answer? Suppose a B, 1 and 4. She's correct. So recognition threshold is basically the minimum concentration of any stimulus that allow the individual to correctly identify something. So for example, the concentration of sugar at which a person cannot even detect it, the sweetness. So sugar should be in the minimum concentration that it can be recognized. So that is the recognition threshold. So basically to determine the recognition threshold in the sensory test, we should have which practices? We should have first and fourth, which we can use the forced choice method and avoiding the distraction in the sensory environment. So a forced choice method basically is effective for determining the recognition threshold because it requires personalists to choose the correct sample among options, providing a structured and objective way to measure when a stimulus is correctly identified. One of the sample contains a stimulus at various concentration, helping them to pinpoint the detection threshold. So this method reduces the guesswork and enhances the reliability of the data collection. Avoiding distractions in the sensory environment is very crucial as external stimuli like noises and the odors can interfere with the personalist ability to focus and accurately perceive the targeted stimulus. These practices ensure the threshold is determined precisely and without the external influence, whereas the detailed background information can result in the biasness and affect their perception. While starting with a concentration well above the expected threshold is inefficient and may skew the results by influencing the person's expectations or causing the sensory fatigue. So, answer first and fourth is correct. Second and three is not correct because they results in the more biasness. Which of the given option are correct with respect to advantage of using electronic tongue in the sensory evaluation? High sensitivity to a broad range of chemicals. Second is effective for solid food samples. Third is permanent data storage for analysis. And fourth is eliminates temperature and humidity effects. Anyone who would like to answer this question? Osia C, first and third. High sensitivity to a broad range of chemicals and permanent data storage for analysis. Okay, let's see. She's correct. Basically, electronic tongues, it basically is the in vitro technique that basically mimics the human taste system. So it mimics basically human taste system. So there are different taste areas. So in human tongue, there are the different sensitive areas which are sensitive to different tastes. So here we can have electronic tongue which can mimic those tastes and provide the effective alternative method to the human sensory evaluation while avoiding associated ethical concerns. So it is basically used for recognition of the compound classification and quantification of the compounds from the liquid samples only. So basically, it doesn't take the solid samples, it takes only liquid samples. It imitates basically human taste perception. Then signal is basically being generated based on the interaction of the sensor molecule with the electric potential. Also, like then the signal is being produced and compared, and finally the data is being evaluated and interpreted. So electronic tongue is basically a sophisticated analytical tool which provides high sensitivity to a broad range of chemicals, allowing it to detect the subtle differences in the taste components like sweetness, bitterness, and umami. So which is making it highly valuable in quality control and product development. So it is very sensitive to broad range of chemicals. Another advantage is the capability of permanent data storage, enabling researchers to archive and analyze data over time, ensuring consistency and traceability in sensory analysis. Permanent data storage for analysis is also true here. However, it is not effective for solid food because e-tongue is specializedly basically designed for liquid or soluble substances. And e-tongue basically is sensitive to the environments like humidity, temperature, so it can affect the sensor's performance and data reliability. So it does not eliminate these effects. So these effects will going to hamper the analysis or affect the analysis. It is e-tongue is basically easy to install and it is basically inexpensive, and it detects basically the toxic compounds like the herbicides and the pesticides also, which humans cannot um, taste, human tongues cannot taste because of the ethical concerns, and it provides only qualitative or sometimes semi-quantitative information. So C is correct here, both 1 and 3. Okay, we have in first column different sensors like potentiometric sensor, which is there in e-tongue, voltammetric sensor, bioelectric sensor, and impedimetric sensor. So here in column 2, the primary functions are given like measures current resulting from oxidation and reduction reactions. B is measures electrical potential between the two electrodes, and C is detection of the changes in the impedance of the sample, and D is uses biological elements to generate electrical signals from biochemical reactions. So we need to match here column 1 with column 2. Anyone would like to answer this question? Fosia A. Okay. Anyone else? Okay, let's see. So she is correct. So in electronic tongue, we have different sensor arrays like optical, electrochemical, mass, and enzymatic or bioelectric sensors. In the electrochemical based sensors, we have potentiometric, voltammetric, amperometric, impedi impedimetric and conductimetric. So it based on the potential difference, voltage difference, and it based on the current. Impedimetric depends on the impedance, and conductance based on the conductance is a conductimeter. And based on the enzymatic or biochemical elements, this bioelectric is there, sensor is there. Optical is related to light, and mass-based sensors are also there. So each type of sensor in the electronic tongue, they have the specific function that contribute to its ability to analyze these liquid samples. So in potentiometric chemical sensors, it measures basically the electric potential difference between the two electrodes. Like we have the reference electrode and we'll have the working electrode, and there is this the change in the potential gradient will be measured through this meter, and we'll have some analytical solution. And this is the ion selective membrane. So it basically measures the electrical potential difference between the two electrodes, a working electrode and a reference electrode, without passing any current. So here no current will going to pass, which is useful for determining the ion concentration in the sample. So potential of the electrode E can be expressed using the Nernst equation equals to E naught. So E is basically the voltage difference for electrochemical cell composed of the ion selective and reference electrode. R is the universal gas constant, which is 8.314 joule per Kelvin. F is the Faraday constant, which is 96500 Coulomb per mole, and N is the number of the electrons. Ln AI is basically activity of the principal ion, and E naught is basically the standard potential, and T is the temperature basically. So all these equals to E naught, which is the standard potential, like the potential of the electrodes is equals to standard potential plus RT by NF ln AI. So the different parts in the potentiometric type of this chemical sensor, this different parts are: one is the working electrode, which is made from the inert material like gold, platinum, or the glassy carbon, and it serves as the surface for redox reactions. So its surface area should be in mm square, few mm square, to limit the current flow. Then reference electrode will be made up of the AgCl electrode, like this AgCl wire. AgCl wire is there, and here is the electrode, which is KCl saturation or 3 mole of the KCl, and this is the lipid membrane here. So it measures the working electrode potential and maintains a constant electrochemical potential when there is no current is flowing. So here is the working solution, and here in this there is this electrode. In this electrode there is reference electrode, working electrode, and auxiliary electrode. So based on the potential difference, it is measured and the data is taken in the computer. So the auxiliary or counter electrode is basically a stainless steel conductor that completes the circuit, allowing the current to leave the solution through it after enters via the working electrode. Next is the voltammetric chemical sensor. So it looks like this. So it measures the current produced between the working and reference electrode in the electrochemical cells. So after the initial measurement, a potential basically is applied to the working electrode. So this is the working electrode. Here potential difference is basically detected. So is applied to the working electrode to detect the current generated from the oxidation and redox reactions. So in potentiometric and voltammetric, there are basically redox reaction happening, and based on that voltage is being generated. So this is the counter electrode, and this is the reference electrode. So the current basically is generated from the oxidation and the reduction reaction as a function of the applied voltage. So how much voltage we are applying, based on that there is a redox reaction take place at the electrodes, and based on that we'll get some information on the electrochemical properties of the analyte. So based on that we can understand the concentration of the target analyte, and we can basically then correlate with the measured current, and we can understand, okay, so this is the basically the signal we are getting. Next is the bioelectric sensor. So they are basically the electronic sensors that uses the biological elements like enzymes, receptors, antibodies, or cells. So they are the biomaterials here, like the cells, enzymes, tissues, receptors, and antibodies, and the sample will be in touch of them. So some kind of biochemical reaction will take place, and the same signal will produce, and that signal is captured. So these will generate electrical signals when there is any biochemical reactions occur and allowing the detection of the specific compound in the complex mixture. It works on the principle of interaction of the biological element with the sample as a result of series of biochemical reactions like the enzyme substrate reaction will take place that initiate the transport of electron, which will be taken as the electrical signal. So mainly oxidase, dehydrogenase, these reactions, biochemical reactions, they will going to take place and produce the electrons to detect through the electrical signals. Last is the impedimetric sensor. So when the sample is going to touch this or place on this here, the sensor will going to have some change in the impedance, and the sensor will sense. So it detects the change in the impedance or the resistance of a sample when which can be affected by the presence and concentration of various substances, making it valuable for the characterization of the electrical properties of the liquids. It can be done at one fixed frequency or the broad spectrum using the impedance spectroscopy. So basically, we have these, we discussed about all these sensors here. So here potentiometric sensor is basically related to the measure the electrical potential difference between the two electrodes. The next is the voltammetric sensor, which is measuring the current basically when there is redox reaction takes place. So it is based on the current, and potentiometric is without current. And bioelectric sensors, they are basically uses the biological elements to generate electrical signals from biochemical reactions, and impedimetric sensor, it detects change in the impedance of a sample. So answer A is correct here. Then next is the we have column 1, we have the novel techniques or the methods like electric nose, electric tongue, eye tracking, and fourth is acoustic sensory evaluation. So in column 2 we have sensory evaluating sensory attributes. So here we have their limitations in column 2, like sensitive to temperature and humidity variation. B is only effective for liquid sample analysis. C is requires complex circulatory and is energy consuming. D is limited ability to naturally interpret the visual perception. So we need to match with the limitations. Anyone who would like to answer this question? B, Fosia. So B is this. Okay, let's see. She is correct. So electric tongue, we already just now we saw about the electric tongue, that it is basically used to analyze only the liquid samples, not the solid samples. So it is its limitation that it is only applied like analyze the liquid samples or the emulsions. So electric nose only applied to, sorry, E tongue. E tongue is related to P, which is only effective for the liquid samples. So we have electric tongue, which is related to the only analyzing the liquid samples, not the solid samples. Then electric nose we have, which is the mimicking the human olfactory system or human smell system. It is going to mimic. So it detects and discriminates among complex odors using a sensor array when it is exposed to the volatile molecules of known odors and generates fingerprints of each odor. So these fingerprints are then going to construct a base database and train a pattern recognition system which further classifies and identifies the unknown odors. So we have the this kind of some smell will be there. So in human, the olfactory receptors from olfactory bulb, it will go to brain and the output will be generated. Whereas in the sensor, electronic nose sensor will take it and will produce some kind of the response or some kind, it will sense and produce some kind of the signals, and then data will be going to analyzed, and for the particular order or particular smell, it will generate have some fingerprint. Based on that, we can understand from the database that this is this order. So in this, there is first sample delivery system. So it enables basically the generation of the volatile compounds to of the sample. So the system basically will take the volatile compounds and detect through the eNose. Then detection system. So detection system will have the electronic properties of the sensor, and it can have the volatile uh, compound. It will going to absorb the volatile compound and it will going to generate some electronic signals based on that. And in computing system, what it will do is those responses from the sensor will be recorded and the data will be analyzed or computed based on the statistical models. So basically this eNose is sensitive to the environmental conditions such as the temperature, humidity, pressure and gas velocity, which can impact its accuracy of the odor detection. So it has complex circuit system basically. So it is basically energy and time consuming also, and it provides basically a fingerprint response to the specific volatiles which going to mimic the human olfactory system, and is basically inexpensive and less time consuming, less bias and more consistent. So it's the advantage, but it is having disadvantage that it is affected by temperature, humidity, pressure and gas velocity. Also we can have different sensors in the electric nose, which are metal oxide sensors. So where basically the gas molecules will going to absorb. So absorption of the gas molecules provoke change in the conductivity, change will be there. So this conductivity change is the measure of the amount of the volatile organic compounds which are going to absorb. So these metal oxides, they will going to absorb the gas molecules and conductivity property it will give. Next is the conducting polymer. So it basically depends on the change in the electrical resistance when there is the absorption of the gas molecule. Quartz crystal microbalance measures mass per unit area by measuring the change in the frequency, and it can be stored in database for the future reference. And surface acoustic wave, it modulates the surface acoustic wave sensed by a physical phenomena. Oh, okay, so there are these kind of four sensors in the electrical nose. Then is eye tracking technology. So in eye tracking technology, it uses the sensors to detect the eye feature. So and estimate where the consumer is looking. So where consumer is looking and what are the eye features it will detect. So since human people basically constantly they are going to in the motion. So the visual perception is naturally difficult to interpret. So basically this technique operates through the red eye effect, where the infrared camera going to track the light reflected from the eye to identify the key features. So there is infrared LED direct light into the eyes, and this from the basically infrared LED light, it will be on the eyes of the in the human eyes, and it will basically going to that way it will going to track the human eye and our human pupil and try to understand. So the infrared LED directs basically the light into the eyes and the reflecting of the retina and making the pupil appear as a bright defined disk. So a small glint is also created by corneal reflection by measuring the distance between the pupil center and the corneal refraction, and using this trigonometric calculation, the point of regard is accurately determined. So it is basically going to uh, track how the human eye is seen. So basically the limitation of this technology is the limited ability to naturally interpret the complex visual perceptions because it focuses solely on tracking the eye movement and does not provide insights into cognitive process or understanding of visual contents. So basically it has disadvantage like demerits like cultural variability can lead to interpretation of emotions and complex data interpretation softwares that requires experts to accurately interpret and apply a meaningful way, and requires costly software and equipment and may raise privacy concerns. But advantage is that it provides an objective assessment of the consumer emotional response, captures and analyze facial expressions in real time, suitable for unconscious responses, and versatile in application like can be applied in food product and lab based sensory panels to in store testing can be used. And next is the acoustic sensory evaluation. So it requires complex circuitry and is energy consuming because it involves analyzing the sound property. So it will analyze the sound property and amplify and filter and take the data such as popping, crackling, crunchiness or crispiness within a food. When a food is bitten or chewed or handled, so it will record the sound through sophisticated equipment, making it a challenging to implement in some settings. Main principle is during food is being consumed, there is an action like biting, chewing. It will generate some vibrations within the food structure, which in turn produce some kind of the sound waves. So this sound waves will have the type and the intensity that will be directly linked to the textural property of the food, and that acoustic sensor or the microphone will going to capture these sound waves by analyzing the frequency, amplitude, and duration of the wave. So based on that, the sensory property is being analyzed. This was our last question. So in this question, we have the electronic tongue, which is only suitable for the liquid food samples, electronic nose, sensitive to temperature and humidity variation, eye tracking, limited ability to natural interpret visual perceptions, and fourth, acoustic sensory evaluation requires complex circuit system and is basically energy consuming. So the answer B is correct here. The topics we covered today basically involved the sensory organs and their roles and the uh, five sensory uh, things like or the organs and based on the five sensory organs, the different sensory modalities. And in tactile, we discussed basically in more detail. And then sensory impact on the consumer behaviors we discussed, and we discussed about the anatomy of the tongue or the nose and how it functions and how the perception of the smell and taste is being generated. And we discussed about the adaptation in the taste. And then we discussed about the different sensory evaluation methods like descriptive. Then in we discussed about discriminative, and we discussed about the triangle test and other tests like duo, trio, ranking based tests, then paired comparison test, preference tests we discussed. Dis then we discussed about the psychophysics of sensory perceptions, about the threshold, limit detection, and what are the different uh, sensory parameters which are affected by different factors. Then we discussed about the electronic nose and electronic tongue, and we discussed about eye tracking techniques, acoustic techniques. And in the next session, we'll be discussing about water and ice, sugars and oligosaccharides, starch, cellulose and pectin, protein and polypeptides, then triglycerides and phospholipids. So this lecture you will get on YouTube, recorded lecture, and the PPT will be uploaded on this drive. And if you have any question, please go in Q&A and ask, feel free to ask question. Or if you have other, like if you have some doubt right now, you can ask me now. Thank you so much for joining the session. So let's meet on next uh, Sunday, same time 4 to 6. Thank you so much for joining the class. So if you have any doubt, please feel free to ask, or otherwise you can leave the meeting. Thank you so much for joining the class. And if you don't have any doubt, please, you can leave the meeting.